So, prokaryotes, as their name implies, are organisms that do not contain Okay, so people are still asking about the antigens. So before I move on to prokaryotes, let's talk real quick about those antigens. If you are A-type blood, on your red blood cells, you're going to have antigen A. If you are B-type blood, you're going to have antigen B. Okay? What that also means is people with A-type blood are going to make antibodies against B-type blood. People with B-type blood are going to make antibodies against A-type blood. People who have AB blood don't make any antibodies, and people with O-type blood make both A and B antibodies. Okay? So if you have A-type blood, you have the A antigen, but you make antibodies against B-type blood. If you have B-type blood, you have the B antigen on the surface, but you make antibodies against anti-A, because your blood can't accept A-type blood if you're B blood type, and if you're A blood type, you can't accept B-type blood. Okay? All right. So, prokaryotes. Here we go. With prokaryotic cells, what we're looking at are cells that do not have a nucleus. No nuclear-bound DNA. Instead, we're looking at bacteria, and I'm just going to draw you a general picture of bacteria. Okay, this is just a generic picture. Bacteria all have cell walls. They've got a plasma membrane. They're going to have DNA contained in a region called the nucleoid. They're going to have ribosomes to produce proteins from that DNA. And finally, they're going to have small circular pieces of DNA they can take up from their environment, and they can actively express genes from these pieces of DNA. And these pieces of DNA are called plasmids. Okay, so there are two major things you did in this unit. One was identifying bacteria based on their shape or morphology. The other was identifying them based on their staining within a gram stain. So let's first start by classifying bacteria based on morphology. Okay, so classifying them based on their shape. Okay, so morphology of bacteria. There are three classifications. Coccus, or cocci, bacillus, or bacilli, and finally, spirillum, or spirilla. I'm going to give you an easy way to remember each of these morphologies. Coccus and cocci look like this. They are circular. So coccus or cocci start with a C, and so does circular. Circular means it's coccus or cocci. Bacillus are typically going to have a shape like this. Okay? They are bar-like, brick-like, or some students like to say they're bratwurst-like. In any case, they all begin with B. So bacillus or bacilli are bar-like, brick-like, or bratwurst-like. And finally, spirilla are snake-like or spiral-like. So spirillum or spirilla are snake-like or spiral-like. So that's classifying bacteria based on morphology. If you see any slides of bacteria, you should always immediately begin identifying them based on their shape, based on their morphology. You should be able to say, this is coccus, this is bacillus, or this is spirillum. All right, so that's morphology of prokaryotes. Now let's talk about the stain. All right. So when we talk about the stain, we're going to be specifically looking at a particular type of staining procedure called the Gram stain. And in this staining procedure, we're differentiating between two cell types, cell type A and cell type B. Cell type A has a very, very thick peptidoglycan cell wall. If you think about what that word peptidoglycan sounds like, it sounds like peptide and glycan. Glycan means sugar, peptide means protein. So this has a very thick peptidoglycan cell wall, while cell type B has a moderately thin peptidoglycan cell wall, but it has an outer layer or structure called the LPS. LPS stands for lipids and polysaccharides. So LPS layer stands for lipids and polysaccharides. This has a thin peptidoglycan cell wall. Okay? But it has that outer LPS layer. All right, so our staining procedure. There are 4 primary stages of staining after you've heat fixed your bacteria to your slide. The first part of the staining procedure is going to be to add a substance called crystal violet to both types of cells. So I want you to remember the relative structures of these 2 cell types. Okay? So let's go through the protocol together. Here we go. So, I've got my first stain of my stain protocol. And the first thing I'm going to add to these cells is a substance called crystal violet. As its name implies, it's a violet dye. And what it's going to do is it's going to permeate the peptidoglycan cell wall through pores, and it's going to wind up in the cell wall or inside of the cells. So it's going to turn these cells purple. Okay? Cell type B. Crystal violet's going to penetrate and get in between the space of the LPS layer and the peptidoglycan cell wall. So it's going to also stain these cells purple. So right now, no big difference between these 2 cells. Okay? So we've stained both cells purple. Next, I'm going to add a substance called Gram's iodine. Gram's iodine is not used to stain the cells. Instead, what Gram's iodine does is it interacts with crystal violet. And when it interacts with crystal violet, it forms the crystal violet iodine complex, which is a huge complex compared to either crystal violet or iodine by itself. So it makes this complex, and this is going to come into play in one of the later steps. So right now it doesn't change any of the coloration of the cells, and both cells remain purple. Okay? So all I've done now is added iodine to make the crystal violet iodine complex. The next thing I'm going to do is I'm going to de-stain. I'm going to decolorize, which I do using 70% ethanol. When I do that, two different things happen. In cell type A, the peptidoglycan cell wall dehydrates. And this results in the cell shriveling up and trapping the crystal violet iodine complex inside of the cell and the cell wall. These cells remain purple. Cell type B, though, doesn't do the same thing. Instead, the ethanol from the decolorizer causes the LPS layer to become disrupted. When that happens, all of the crystal violet iodine complex is washed away with the LPS layer. And these cells have no color. They're colorless. So far, so good. But the problem is, if we're looking at cells under a microscope that fall into the classification of B, there's no way for us to see them right now because they're colorless. So the final thing we add is a counter stain. And that counter stain is called safranin. It's called safranin. The counter stain is called safranin. And safranin turns these cells, well, it doesn't have any effect actually on this first cell type. So this cell type here, A, move my counter stain here just a little bit. This cell type here for A remains purple because all of the pores and the cell itself are occupied by the crystal violet iodine complex. But cell type B is stained red or pink. So we now have arrived at a difference between these two cell types based on the outer structures on the cells. And we classify cell type A as gram-positive cells. Cell type B, on the other hand, are classified as gram-negative cells. Does that make sense to everyone? So gram-positive and gram-negative cells. Questions pertaining to gram stain and morphology? If not, as soon as you guys are ready, we'll jump into mitosis and meiosis, and then we can call this done.